Welcome to everyone from around Australia, and I think we have some international guests as well, so welcome. My name is Helen McGuire, and along with Fabio Luciani here, we will be your hosts for today. I'm a researcher at the University of Sydney, and I'm interested in single cell technology. So if you're on the same page as that, you're in a great place today. So I would like to begin by acknowledging the traditional owners, traditional custodians of the land on which we meet. So today I'm sitting at the University of Sydney, which is the lands of the Gadigal people of the Eora Nation. And I pay respects to the elders past and present. So I also want to extend my respect to the traditional owners of the lands on which you are joining from today, because it is in this virtual setting that we're all here today. So it's really nice. So for those of you, if this is your first OzSingleCell experience, a warm welcome to you, of course. We are a grassroots organisation, and we're inspired scientists around the Australian community, of which put technology, biology, molecular biology and computational, so all scrambled together and with a single cell interest. We're all centrally themed around biology. So today our biology is immunology that we're going to be discussing. But first I'd like to take us through a few housekeeping and we'll get to that first. So you're joining us here on a Zoom webinar. It's our alternative to what we could do this year, which was not join for a regular conference. So today, this is our 8th episode of the series, which we keep extending, but we'll finish out at the end of this year. We think it's a highly successful initiative. We're thrilled to bring you the content so far, and we encourage you to revisit the previous episodes that we've prepared so far, and these are available on demand at singlecells.org.au. So it goes without saying that an ambitious venture such as this series would not have been possible without our amazing organising committee. So we've had a whole bunch of people behind the scenes to work very hard on this, and it's been an excellent and evolving program. So Christine Wells, Kwan Nguyen, Heather Lee, myself, Manu Singh, Joseph Powell, Alicia Oshlak, Al Forrest, Fabio Luciani, Daniela Zaliskstein, and Charlene Naik. So it's with this amazing support of, and we also had Helen along the way. So with this, I encourage you to visit our website, and also I will just take this opportunity to thank our series sponsors. So we certainly could not have done a promotion such as this without all of our wonderful sponsors here. So many thanks. And I think we can all agree without any second guessing, this could not have happened without your continued support. So today I mentioned I'm here with Associate Professor Fabio Luciani, and I did want to take the opportunity to introduce Fabio because, as a co-panelist here, he's also very well known in the single cell space as well. So he's a research fellow at the Immunogenomics at the School of Medical Science and the Kirby Institute at UNSW. He is trained as a physicist, theoretical physicist and an immunologist in Australia. And he's moved to Australia that he's led a mathematical modelling program in infectious disease, but today he really focuses on multi-omics research. And it's a study of T-cell responses in the context of viral immunity, viral infection, autoimmunity and cellular therapies, which is really playing in the multidisciplinary space that he contributes. We'll look forward to his contributions in the discussions in today's episodes. So I did say we had some housekeeping. Really simple. We've set aside about an hour and a half to have some really great presentations and a really interactive Q&A session at the end. And so with that, I will point you to the Q&A function of Zoom, which you may be familiar with already. We've enabled the upvote function. So if you see a question in there already that you really like, certainly go ahead and promote it a little bit further. And our moderators, we will filter these and get some of these along the way answered. So if you're also interested in Twitter, we've indicated here a couple of hashtags. We'd love you to continue the conversation after this, brief time that we can present online today. So OzSingleCell and single immune cells. So hashes in front of those. So our episode today is brought to you by BD. And for those of you unaware, our first speaker is from BD, so we'll get to hear a little bit about that. And to kick us off, I will invite Fabio to continue the introductions. Thanks, Fabio.
Thank you, Helen, for these kind words, for everything you have said. It's wonderful. Welcome, everyone. This is a very exciting day for me because it's the first immunology application. So the application of single cell to immunology. And I'm always very excited to present these things because immunology is, in my opinion, is one of the greatest places to really see the power of single cell genomics. As all of you know, I don't want to spend too much words, but this is really a revolutionary technology, has really changed a lot of things that probably are likely to end up in the next textbooks of many, many biological fields, including immunology. And the simplest thing to do, immune system moves over time, right? Responds to viruses. Just look at what happened with coronavirus. And if you quickly PubMed, it's insane the amount of research where people have intuitively just said, let's do single cell genomics to understand what the immune response is to these viruses. And I think this really manifests how important is this technology in immunology. A couple of words I want to mention on, I think Helen has mentioned that we've been playing with single cell genomics for a while. Australia's been one of the early adopters, as we discussed in the last webinar we had, where we have a bunch of people talking about this. And it is true, we evolved quite rapidly. And, I'm always very proud, even if I'm half Italian, half Australian. We had several Nobel Prizes in immunology. Always the question, where how new cell types, how these T cells and B cells evolve, the monocytes. And I think this gives us a very great feeling of how we are probably going to speed up even more rapidly than now and help especially, patients in translational application of this. So I don't want to spend too much more time about these words. We can discuss a little bit later. I would love to introduce the first speaker of today is Maggie Nakamoto. Maggie joined BD in 2018, and she actually now leads the genomic application group within the single cell multiomics field of BD. Her team contributes by developing technologies and also application of a very novel platform that BD has, which is the BD Rhapsody. And Maggie has a PhD in RNA biology from Stanford University in 2017. And I've been knowing actually personally Maggie, I think for 2 years, where we did some work together. And I'm a little bit aware of the type of work that she is doing. And it's not only actually one modality, she actually performs a lot of work on single cell multiomics. And as you can see from the title that just popped up now, she's going to talk to us about driving deeper biological insight and single cell multiomics analysis. So with that, I would like to thank Maggie for joining us today, and please tell us your stories. Thank you.
Thank you so much for that introduction. And yeah, I just want to start by thanking you all for inviting me to chat here. I loved OzSingleCell 2018 in person, and this seminar series has been really fantastic so far. So I'm just really excited to get a chance to share some of our stuff here as well. So as Fabio said, my name is Maggie Nakamoto, and I manage the single cell multiomics applications group at BD. So we do technology and application development for the BD Rhapsody. And I'm going to talk a little bit today about the Rhapsody platform for anyone who's not familiar with it. But as Fabio said, what I really wanted to do was showcase some of the multiomics analyses that we've been working on. So in single cell, I think a lot of us now are interested in looking at molecules beyond just RNA. And so today, as I said, I'm going to talk about the Rhapsody, but ultimately I want to tell a little story showcasing how we kind of bring together RNA and protein, and in this case, TCR, clonotype analysis, to kind of get a deeper look at the biology going on. I will say we're obviously mostly doing technology development. So the system we're using is a little contrived. View it as a warm-up for the next talk where you'll hopefully be seeing some really cool biology. But I hope at least I can show kind of how we're using this multi-omics technology. So yeah, so for anyone unfamiliar with the BD Rhapsody system, that's what I'm going to be talking about today. And this is really, we're trying to make kind of an end-to-end solution here. We use the Rhapsody itself for kind of single cell isolation and barcoding, and then we have a number of different library prep options to look at different types of molecules in the cell. And then we have a number of data analysis tools at the back end. I won't really be talking about those today, but I do want to introduce the machine itself for anyone who hasn't, who's not familiar with it. So this is the Rhapsody. It has two components. There is a scanner on the left, this larger instrument. I'll come back to that in a minute. But on the right here is what we call the Express, and this is really where the action happens, because this is where we actually manipulate the cartridge, which is what we use for our single cell capture. So right here, if we zoom in, this is what we call our cartridge. It's really simple. There's a little port here that we pipette into. Liquid flows through this U-shaped channel, and it flows out through an outlet port right here. And if we zoom in on this U-shaped channel, what we'd see are hundreds of thousands of these microwells. So basically we can flow a single cell suspension through this cartridge, let the cells settle into the microwells. We then flow magnetic beads into the cartridge, let those settle as well. And once we have a single cell and a single bead paired in the cartridge, we just flood the cartridge with lysis buffer, and our cells lyse open. That's what's depicted here in the dotted orange line. Our cells lyse open and our polyadenylated mRNAs come out, and they hybridize to our barcoded cell capture beads. We then recover the beads simply by sticking a magnet on top of the cartridge. I mentioned the beads are magnetic. So we just pull them up out of the wells and flood it with buffer and recover all of those beads. So this is a really simple system. There's a couple of magnets on it that you use to help pull beads down into the wells or up out of the wells. And there's a little slider on the front here that you use to access your waste, your beads after you're done with your whole workflow. But there's no microfluidics, no electronics. It is very, very straightforward. So that's, again, all you need to actually do the capture. I did say I would come back to the scanner. And so the scanner is actually a QC device, but it can be somewhat powerful. So basically what it does is it scans the bottom of the cartridge at each step of the workflow and gives you some information about how you're doing at each step of the process. So this is an actual image from the scanner. You can see the wells here. The dark spots in the wells are our beads, and the green spots you can see here are cells. So at each step of the way, your scanner can tell you how many cells you captured. It can tell you about the viability of the cells. The cells are green because they're actually stained with a viability dye. It can tell you about your multiplet rate. You can actually see right here a doublet. And then as you move into the bead loading and bead retrieval, it can tell you, how successfully you've loaded beads, how successfully you've retrieved them. So this can be really important, especially as you're making decisions downstream about how much money you're going to pay for sequencing, whether you've captured enough cells to move forward with the process, things like that. Before I move into the assays, I just want to talk for a minute about this microwell system. We really love it. We think it's quite powerful, and we see, first of all, really amazing consistent performance. These are just gene by gene correlation plots looking at expression of genes in here on the left, technical replicates. In the middle, you're looking at user to user variability. On the right, we're looking at site-to-site variability. So these are the same PBMCs, but they were shipped to 2 different sites. So these are different users, different days, different Rhapsody instruments, different sequencing runs, and with no batch effect correction, we already see really beautiful correlation of the data. So we really do see this very consistent performance, and we think that's partially because, you know, with this microwell system, we're able to see very faithful capture of different cell types and very faithful encoding of RNA onto the beads of cDNA. So just to showcase that, this is an experiment we did. We put in, first of all, cells of different sizes, so small, medium, and large cells. We used Jerkit, K562, and BT546 cells. Not a huge range of sizes, but we're at least getting some spread there. And then the other cell type we loaded in here was neutrophils. And we were interested in granulocytes specifically because they can be very challenging, especially in closed systems. You know, after you lyse up in those granulocytes, all of those kinds of toxic effector molecules are in that environment where you're trying to perform reverse transcription, and that can be really challenging. So we kind of wanted to see how they perform in the Rhapsody. And what we saw was really, again, great performance of the system. So in orange here is the number of cells loaded for all of our different cell types, and in blue and gray are the cells retrieved by sequencing in 2 replicates. And so we saw, as you can see, nice reproducibility, no kind of bias in terms of cell size that was being recovered, and really beautiful recovery of those granulocytes. We think that's because, you know, we lyse them open in that microwell, but then we can recover the beads and wash them and perform reverse transcription in a really happily buffered environment. So that really does help with very faithful encoding onto the beads of the molecules. So with that open well system, though, there can be some concerns about things like noise in the system. So we did want to, you know, highlight or generate some data to kind of speak to that. And so in this experiment, we loaded 40,000 cells into the cartridge. We didn't want to mess around with 1,000, 2,000 cells. It's easy to not see any crosstalk with those kinds of cell loads, but we wanted to push up to 40,000. That's something we can do pretty easily with our cartridges. And we did a 1-to-1 mix of human and mouse cell lines. And here I'm just showing mouse molecules per cell on the x-axis and human molecules per cell on the y-axis. Each of these dots is a cell. And we see, even with that really high cell load, really beautiful data. Not really seeing any human molecules creeping into our mouse data, and vice versa. They're laying really cleanly kind of right on the axis. And in the middle here, you can see our doublets, which is kind of the last thing I wanted to mention about the microwell system that we love, which is the really low multiplet rate. So again, we're doing 40,000 cells here. The theoretical multiplet rate based on Poisson would be about 9%. With our scanner, we're spot on, right at 9%. I also threw in doublet rate based on phenotype analysis, both mouse-human and human-human or mouse-mouse doublets. And again, slightly bigger spread, but still right around that 9% mark. So, you know, with these microwell systems, we're seeing really consistent capture. We're seeing very low noise and very low doublet rates. So that really helps us be confident that the data we're getting is clean, and we're really maximizing, you know, the number of cells we're getting usable data from. Okay, so I want to move forward into talking about the assays so we can talk about the data. I'm happy to chat more at the end about the system if there are any more questions. So moving forward, obviously, once we've captured our library and have our cDNA library on beads, moving forward, there are a lot of different molecules to look at. So I'm going to talk about a few of our assays today. On the RNA side, we have 2 options. The first is targeted RNA-seq panels. So these are sets of 400 to 600 genes related to specific processes that you might be interested. We have 4 off-the-shelf panels ready to go, and you can supplement up to 100 more genes that you might be interested on top of those panels. And then last year, we also launched a whole transcriptome amplification kit for people looking to do kind of an unbiased just look at the transcriptome. It can be really good for biomarker discovery and things like that. The other thing I wanted to highlight briefly, because I'm going to be chatting about it in our data, is our BD-ABSeq antibody oligoconjugates. So this is basically a way that you can look at protein on the Rhapsody system using sequencing. So for anyone who's not familiar with these, basically for oligo-conjugated antibody technology, what we do is we take antibodies that already exist in our BD catalog, and instead of conjugating them to a fluorophore, we conjugate them to a DNA oligonucleotide. And that oligo has a few key features. The first is a barcode that tells us which antibody that oligo came from. The second is a poly-A tail that allows it to be captured the same way we'd capture an mRNA in our cartridge. So the Rhapsody workflow remains pretty unchanged. Before we load the cells into the cartridge, we stain them with a panel of ABSeq antibodies. But then we go through loading and lysis, it's all the same. When we lyse open our cells though, in addition to the mRNAs hybridizing to the beads, we're also gonna see those AB oligos hybridizing to the beads. And when we do library prep, we're gonna have a library of these AB oligos that we can use to get information about protein expression. So that's kind of the antibody oligo technology in a nutshell. And then the third omic I'm going to be talking about today is TCR-BCR clonotype analysis. So this is a protocol that we launched at the end of last year that allows you to profile specifically the CDR3 region of T and B cells. We'll have a kit coming out next year where you can do full length, but for today I'm just going to be looking at CDR3 only. So for those in the room who already know TCR and BCR technology, you're all familiar with the challenges of getting this information in single cell. For those who aren't familiar with it, the challenge is really the fact that this VDJ information we're interested in is located all the way at the 5' end of the transcript. So this is really not drawn to scale, but just to give you a picture, you know, in a 3' RNA-seq or in a 3' capture single cell assay, we have our cell label down here, and our VDJ sequence is encoded onto the bead. And what we're interested in is all the way here at the 5' end. So this whole thing is, you know, many thousands of bases long. So in order to get this information, this VDJ information alongside a cell label, you either need to do very, very long rate sequencing, which is definitely one option. The other option is to bring this VDJ information closer to your cell label. So the way we did this with Rhapsody is some kind of fun, I think, molecular gymnastics. We basically use template switching to add a new capture sequence onto the 5' ends of the bead. And then what happens is that capture sequence comes down and it hybridizes to oligos on the beads, and we then get a cell label added to the 5' end. So the ultimate molecule that we have is our bead with the original cell label, the whole transcript, and then an additional cell label on the other end. So we can still amplify from the 3' end that immune response panel that we were originally amplifying. But additionally, on the 5' end now, we can use a VDJ primer panel to also amplify up that VDJ sequence on the 5' end. So with that, I think I've kind of described all the technologies I'm using in this talk. We used the Rhapsody for cell capture. And then for RNA, we used our RNA targeted panels. We also did ABSeq and TCR analysis in this experiment. So this was specifically an experiment where we wanted to kind of stretch the legs of our VDJ assay. And alongside that, we wanted to take a look at kind of T-cell activation. So what we did is we used the human immune response panel, which is 399 genes that are focused on kind of the immune response, and a 20-plex ABSeq panel focusing on activation and exhaustion markers primarily. And this was our experimental setup. So we had PBMCs and we stimulated them repeatedly with synthetic viral peptides. The peptide pool that we used over here was called a CEF pool, and that's referring to the 3 viruses that were used to derive the peptides, CMV, EBV, and flu. And so our experiment was basically stimulating these T cells on day 0, day 7, and day 14 with this peptide pool. On day 9, we looked at the cells with flow cytometry to just monitor T cell activation. And on day 16, we took them through the single cell multiomics workflow. So this experiment, we used 2 different donors. In addition to that CEF stimulated sample, we also had a DMSO control for each donor. And as I said, on day 9, we took them through flow analysis, and on day 16, we took them through the multiomics workflow. We also sorted out CD3 positive T cells at the beginning of the workflow before loading them onto the Rhapsody, so we could really focus in on our T cells. So the first question we had was really, how is our VDJ assay performing? And to get kind of a bulk look at this, we used these chord plots to kind of get a snapshot of the system. So what I'm showing here are pairings between V and J segments. So J segments are along the top, V segments are along the bottom, and the chord between them shows every connection that existed in 3 or more cells. So expanded clonotypes can be seen as thicker chords between specific V and J segments. So on the left here, we have our DMSO control, and on the right, we have our CEF stimulated cells. And so we can see a lot of these kind of thicker chords corresponding to expanded clonotypes showing up in our CEF pool. So just to highlight one of them, this one right here, this dark gray, is TRAV5, TRAJ23. And we can see a really distinct connection between these 2 in our CEF stimulated pool. And over in our DMSO sample, we don't see any kind of distinct connection between those 2. So just to quantify it for you, in our DMSO sample, we saw 5 cells with that pairing. In our CEF sample, we saw 127. So we can see this clone that's specifically upregulated in our CEF stimulated pool. This one we also actually saw expanded in both donors. So this TRAV5, TRAJ23, I can also refer to as a SIGCLE, which is the alpha chain CDR3 sequence. This one is fortunately very pronounceable. So we can see this SIGCLE clonotype. Here I'm showing the % of T cells expressing that clonotype in the 2 donors in DMSO or in CEF conditions. And so we can see it really is specifically expanded in CEF in both donors. So the next thing we did is we went to online VDJ databases, and we checked to see whether this clone corresponds to any known viral peptides. And in this case, we're really lucky. We found that it's known to recognize an epitope from EBV, and this epitope was actually in that CEF pool that we had used. So this is fantastic news. We can see expanded clonotypes that correspond to peptides that were in our CEF pool. This obviously wasn't the only expanded clonotype. Here I'm showing the top 15 expanded clonotypes in the 2 donors. Really reassuring to see that a lot of these clonotypes are specifically expanded in CEF. They aren't seen at high rates in DMSO, with the exception of this guy over here, which might have existed before the experiment or could be responding to something in the DMSO. But the majority of these seem to be really CEF specific. Here's that SIGCLE clone in case you were wondering kind of where it ranks. And when we ran these through the VDJ database, we found a number of clonotypes that are known to recognize epitopes that were in our peptide pool. And we were able to find expanded clonotypes towards all 3 viruses. So this was really fantastic, reassuring for us that our VDJ assay really is, finding some real biology here. But as I said, we also had these 399 mRNAs and these 20 ABS-seq markers, and we really wanted to go back and see what we could do with this multiomic information. So what we did is we used that RNA and protein information to create a TriMap projection to look at the trajectory of the TCR phenotypes. So on the left here is the TriMap projection that was generated. And the cells are colored based on these groups that the program came up with, kind of just based on the gene and protein expression. And so to understand what these different groups were, what we did is we came up with a list of genes and proteins to kind of define different sets of T cells. So naive, memory, activated, and exhausted T cells. So this is a heat map. Each row is a cell, and we're looking at the expression of whichever gene or protein is denoted on the top here. And the cells are grouped together based on the groups that were labeled in the TriMap projection. So to kind of identify what these groups were, I'm not showing all of them here, but for example, group 12 here, what we saw was upregulation of naive markers like CD45RA. And so group 12 here is our naive cells. Moving up into groups 5 and 2 here, we see these cells are starting to upregulate activation markers like CD25, things like granzyme, perforin. Group 2 seems to be a little more highly activated than group 5. So groups 2 and 5 right here, we have our kind of low level activation group and our more highly activated group over here in red. And the final one I wanted to highlight is our exhausted group. So in addition to the activation markers here, these cells are starting to upregulate exhaustion markers like CD279. So at the end here, we have our exhausted cells, and using this, we can kind of trace a trajectory here of kind of bulk level, just naive, activated, exhausted. So once we had that TriMap projection, what we did is we went back and we projected the cells that we had seen having those expanded clonotypes onto that projection. And what we saw was interesting. The clonotypes corresponding to different viruses were definitely found in unique regions. So our CMV cells were really found in that kind of low level activation cluster. Our EBV specific clonotypes were found, one was found in that very highly activated cluster, and then a couple in that kind of exhausted cluster, and our flu expanded clonotypes were in that kind of exhausted cluster. And we can see that as well by just looking at those clonotypes in the heat map. So this is the same genes I was showing before for the activated and exhausted, but now the cells are grouped based on their expanded clonotype status. And so, you know, we can see our CMV specific clonotype with kind of low level of activation, very different from this flu clonotype up here, which is much more highly activated, and both of those in contrast to our old friend Acidcli here, which is starting to upregulate exhaustion markers like CD279. And so ultimately, when we can do this, you know, this was a single experiment, a single cartridge where we looked at all of these different molecules. And so this multi-omic analysis really allows us to say, you know, we can find these TCR sequences. Sometimes we can even identify the specific peptides they're recognizing, and then go back to the RNA and protein information to understand the phenotype of those cells, whether they're kind of at this low level of activation, this flu-specific one that's just pumping out granzymes, perforin, interferon gamma, very activated, and then, you know, other clonotypes like this one over here that's kind of shut down some of those effector activities and is really upregulating exhaustion markers like CD279. So just to conclude, you know, this was fantastic to see that our assay can really identify these expanded clonotypes that correspond to viral peptides that we knew were in our pool. But just coming back to that multi-omic analysis and really thinking about how the ability to look at that protein, that mRNA, can help you see very distinct activation and exhaustion states for your different cell subsets, and really start diving into the biology when you can look at all three of these at the same time. So that's all I have. I wanted to acknowledge the scientists who did the work. I am very happy to have such beautiful data to present, but I am not the one doing the actual stuff in the lab. So I wanted to acknowledge Tim, who did a lot of the experiments at the front end that I showed, showcasing the microwell technology, and then Shirley, Imtiaz, and Christian, who did the work and the analysis on the VDJ data set. So yeah, I just want to say thank you to them. And with that, I'm happy to take any questions about this or anything I talked about today.
Awesome. Thanks, Maggie. That was really fantastic. So I might, while people gather their thoughts, because you've had quite a lot of exciting content there. So I might get started with a question. So am I correct in understanding that the epitopes that you had uncovered were only ever found in one type of phenotype? Like, that's a pretty cool observation. Like, they didn't spread out across the different clusters.
No, we saw them pretty specifically. You mean like each expanded clonotype was found in a very specific area? Yeah, we did see that. So the flu ones were the only ones where we saw like a couple of them, like one main cluster and then a couple cells that would be over in other areas. but for the most part, yeah, they were all pretty similar in their activation states. And I don't have enough immunology background to know if that's kind of what we definitely expect, or you know, I also don't know how much it's because we did this at day 16. So, you know, we're at that point, a lot has already happened with these cells. They've already, we've already moved through a big biology space during that time, and we are definitely at an endpoint, I would assume. And so that could also have something to do with it.
Yeah, that's an interesting concept. I suspect that might be the case. If you were to iteratively sample, you'd see that progression and decisions, and, you know, one avenue being, you know, no further continuation, I imagine. Anyway.
Yeah, that'd be fantastic.
Yeah, one more, just a super technical one from me was, I was just curious more than anything whether you knew the HLA type of your donors, your donor A and B.
I do not. I don't know if I don't, sorry.
Yep, that's okay. So we've got one question from the audience. So Mani Gupta, who's in Sydney, has asked, does the BCR sequencing and Rhapsody, is that able to sequence constant regions? And does it also give you the isotype information like IGHD and IGHM?
Yeah, so we should be able to sequence the constant region just based on with our 3' primers. So our 3' assay has usually given us that general isotype information. Yeah, so now we can do the CDR3 with the kind of constant region. What we still can't do with our CDR3 only is the whole variable region. So we can't get the hypermutation that's happening, and that's what we will be able to get with our kit that's coming out next year. So that's when you'll kind of have the whole picture of the BCR.
Great, wonderful. All right, we'll keep thinking of those questions because we'll take it all to a big Q&A at the end. But in the meantime, I'd absolutely love to welcome Harry Sutton to the stage, the virtual stage as it is. So Harry is joining us from Canberra. He is a PhD student at the the Cockburn Lab in the John Curtin School of Medical Research. His research is primarily focused on B cell responses. So we're going to hear a lot more about B cells today. And this is towards pathogens such as Plasmodium parasite that causes malaria. So in particular, he is utilizing single cell technologies to characterize the poorly understood populations of atypical B cells. And so it's a really nice, you know, his talk here, title is Atypical to Typical, an alternative way to examine B cell responses. So pretty excited to hear all about that. Thanks, Harry.
Okay, thanks, Helen. Thank you guys for having me to discuss this data. I've been to one single cell conference before and it was really interesting. There isn't much single cell things being done at John Curtin, so it's nice to talk to people who actually have a bit more experience in it. And then saying that anybody who's seeing this talk and actually has any feedback on any of the stuff that we're talking about, that would actually be, you know, it's interesting to get people who have more experience in this data's perspective on it. So I'll be talking about B cells, in particular, a type of subpopulation of B cells called atypical cells. But before we can really talk about that, I'll give a brief description of a typical B cell response. So here we have a happy little B cell. If you become infected with something such as, hold on, my anime, there we say you become infected with a pathogen or exposed to an antigen, whether it be COVID, flu, or malaria, the B cells will specific B cells will recognize this antigen using their B cell receptor. They go receive help from specialized helper T cells, CD4 cells, and from there B cells will differentiate into a number of different B cell subtypes. So for example, some become short-lived plasmablasts, which secrete antibody for a small number of days to fight off the initial infection. But more importantly, some enter what I like to call the germinal fitness center, or germinal center, which is where these B cells undergo somatic hypermutation of their B cell receptor and affinity maturation, leading to higher affinity B cells that will leave the germinal center as either long-lived plasma cells that enter the bone marrow and secrete antibody for hopefully a long period of time, either the rest of your life or shorter, depending on the pathogen you've been exposed to, or memory B cells, which can start the whole process up again when you're re-exposed to a pathogen and become a short-lived plasma cell much quicker, or long-lived plasma cell again. But in some infections and in some conditions, there seems to be another type of B cell population which is prominent. And these are these atypical memory B cells, sometimes called tissue-like memory B cells, if you're working on HIV. These cells have been found in people exposed to malaria, people who've been infected with HIV, people who've been infected with hepatitis C, as well as autoimmune conditions. And they're generally considered, because they're found in all these conditions that are sort of related to chronic antigen exposure, or they're sort of considered to be exhausted cells, particularly as well that early studies on them found that they appear to be dysfunctional. Their B cell receptors didn't signal very well, and they don't readily differentiate into antibody secreting cells. So they're sort of thought to be they're originally thought to be a defective population. There's argument in the field now of where they sort of come from. Do they come from naive cells and during the primary response? Do they come from the germinal center? Early studies again found that they weren't very somatically hypermutated, which leads people to think that they don't go through a germinal center. And in fact, some newer studies now, particularly in mice and in some autoimmune conditions, seem to suggest that actually what they are a precursor to short-lived plasmablasts, and that they're this short-lived activated cell. So there's quite a lot of debate in the field about what exactly these cells are and what's their purpose. So to sort of go back to an old school, the classical flow cytometry definition of memory B cells that people used to use, particularly, is this based on two proteins in particular, CD27 and CD21. So if you were a B cell that only expressed CD21, you were generally classified as a naive cell. If you expressed both CD27 and CD21, this is generally a marker of a memory cell. CD27 cells are generally thought to be recently activated, and then will transition to become memory cells or plasma cells. And then if we actually look, so this was all in a non-exposed healthy individual. If we look at somebody who has previously been exposed to multiple episodes of malaria, they actually have quite a large population of these CD27, CD21 negative cells. And these are what have historically been thought of as the atypical B cell population. Although in more recent studies, particularly in flu, they argue that all CD21 negative cells, or most CD21 negative cells, are also related to this atypical cell population. Other common markers of atypical cells include CD11C, FcRL5, as well as TBET. So the FcRL5 is a common inhibitory marker. So these are markers that you normally see used to describe atypical cells previously in the literature. So as I said, that was from a malaria-exposed individual, and our lab focused primarily on malaria infection and immunization. So I'll give you a brief overview of the malaria life cycle to see if you have an idea of some of the things I'll be talking about in this talk. Malaria infection begins after a bite from an infected mosquito that will deposit sporozoites in the skin, which migrate to the blood vessels, which then use the blood vessels to migrate to the liver, where they'll invade your hepatocytes and begin to change and multiply in the form of a merozoite, which then duplicates and bursts out of the liver to infect red blood cells in the bloodstream, causing this asexual life cycle within the bloodstream, which is the primary cause of disease in malaria. And some of these merozoites will change into sexual stage gametocytes, which can be taken up by the mosquito and restart the whole process again. Importantly, there's a couple of proteins that I need you guys to know about to understand this talk. One of them is the circumsporozoite protein, which is found on the surface of the sporozoite stage of the parasite. This is quite a well-studied protein, and it forms the basis of the only currently licensed malaria vaccine, the RTSS vaccine. It's made up of a part of the CSP. And then we also have the merozoite surface protein 1, or MSP1, which is found on the surface of merozoites, as the name suggests. And it's, again, it's thought of as perhaps a useful vaccine candidate for the blood stage infection, and it's something that we'd have a look at as I go on. So with this talk, I'll be hoping to answer two main questions on atypical B cells. One, are they an activated population? Are they a memory population? Or are they actually exhausted B cells as people originally thought? And we also, near the end, will use some of the data that we get from our single cell analysis to answer this question of when do they appear during the immune response. And by answering this question, we hopefully have a better understanding actually of answering the first question. If they appear much later, then perhaps they're much more likely to be exhausted cells, but if they appear earlier, perhaps they can be a more activated population. So the first thing we actually did was we wanted to sequence antigen-specific B cells, so specific to, in particular, malaria antigens. So I did a lot of this work with Rachel A. from Kemri in Kenya, and their group had access to two interesting cohorts. First was a group of children, young infants, who were immunized with the RTSS, which is again made up of that CSP cell, and then blood was taken 6.5 months after the trial ended and 74 months after the trial ended. So we took CSP specific cells from that cohort. We also had access to an adult cohort of Kenyans where we could also access CSP specific B cells, which would have been generated just due to natural exposure of malaria, as well as MSP1 specific B cells, again from natural exposure to malaria. And we were also interested in taking tetanus toxoid specific B cells from this group, as this represents, I guess, a more normal vaccine antigen. Unlike CSP and MSP1, where if you live in a malaria endemic area, you'll be chronically exposed to this antigen. These individuals would have only really hopefully been exposed to tetanus toxoid when they were vaccinated against it. So it's sort of a control antigen. So to identify antigen-specific B cells, we use this tetramerization process. So you take your antigen, you biotinylate it, and then mix it with fluorescent streptavidin. This forms a tetramer, which your antigen-specific B cells can bind to, and then using flow cytometry, you can identify these cells. So we're using two different colored tetramers, and you can see an individual who lived in a low transmission area has very few or hardly any MSP1 specific B cells. However, in a high transmission area, they have more MSP1 specific B cells. So this is how we were able to identify antigen specific cells, which we could then sort. And in this case, we could only ever really find around 20 to 50 antigen-specific B cells per person. So we used the SmartSeq2 single cell method because it's designed for lower input, and we didn't throughput, and we weren't going to have much throughput with this in this experiment. So we sequenced the cells, and using SURAT, we performed unsupervised clustering, and that from around 180 cells in the end. And that revealed three distinct subsets. Although I say distinct, some people probably are more used to seeing, this is from SURAT's tutorial, more used to seeing this kind of UMAP when you're looking at single cell data. But of course, we're actually just looking at B cells. So we're looking at cells that are already quite closely related to each other. And so we're trying to examine much smaller differences than if you were trying to look at differences between T cells and B cells. But if we do look at the differentially expressed genes of these clusters, there does appear to be a difference. In particular, this purple cluster 1 we found was enriched for many atypical genes and atypical gene sets via gene set enrichment. And particularly, you can see here it upregulates ITGAX, which is the gene that encodes for CD11C. This second population we designated as memory B cells. It didn't really have many genes that mapped to memory B cells, but did have some that mapped naive B cells, and naive B cells and memory B cells were quite closely related, as well as having a number of markers such as CCR7, which are common for memory B cells. And then we had this third population, which we couldn't really find out what it was close to, but it does upregulate the chemokine receptor CXCR3, which we thought would be an interesting marker for further study. And again, we were able to look at, for example, what antigens these clusters were found in. So we found, as sort of expected, tetanus toxoid was only predominant. Tetanus toxoid specific cells were predominantly found in this memory B cell cluster, and makes sense as this is sort of this normal antigen, while CSP specific B cells were predominantly found in the atypical B cell cluster. Interestingly, we were expecting this MSP1 specific cells, since it would be quite an abundant antigen during a malaria infection. We thought it would actually induce more atypical B cells if they really are chronically induced by chronic antigen activation. But it seemed to be predominantly made up of cells from the CXCR3 positive cluster. Now, the issue with this experiment is, again, we had around 10 individuals, and we only got around 10 to 15 cells on average for each individual. And we're also looking at multiple antigens. So we were worried about quite a few batch effects with this experiment. So we then wanted to sequence a lot more cells, and we did this using the 10x chromium. In this case, we took 2 non-exposed individuals and 2 malaria-exposed individuals. So these non-exposed individuals from Australia, they've never been exposed to malaria before. And we took their B cells, in particular the IgD negative B cells. So these should be their memory B cells, the circulating memory B cells from the blood. We also stained them with total seq antibodies, so they're very similar to the antibody oligos used by BD. In particular, we used CD27, CD21, CD11C, CXCR3, and CCR7 as the markers we found from our previous SMART-seq experiment. One thing I would say, just in sort of a hindsight thing, initially when we did these, the antibodies were quite expensive and we didn't want to incur too much more cost. But in hindsight, if we were doing this experiment again, and we think we would have added a much bigger panel, because the beauty of these oligo antibodies is you can have quite a large panel since you're not dealing with compensation issues. And so I guess when you're already spending quite a large amount of money on the sequencing, we thought, in hindsight, and just I guess advice to anybody who's thinking about doing the experiments, we would have included more antibodies in this panel if we were able to do it all over again. And we also, so we sequenced the whole transcriptome. We also sequenced the total seq antibodies, and particularly the benefit, I guess, of single cell when you're studying immunology is, particularly if you're studying B cells or T cells, is you can do this VDJ analysis. So we used the Chromium 5' kit, which allows you to sequence the whole VDJ of these B cells to give us just that extra amount of data. So in total, we sequenced 12,000 B cells from the 4 individuals. After downstream analysis, we had around 12,000 B cells, and we got around 11 clusters. So what we're seeing here is a deep heat map of some of the most differentially expressed genes among those clusters. The most interesting things we saw were these sort of 3 related clusters to form the supercluster that we found was enriched for atypical genes. So these seem to be our atypical genes, typical cells. Here we had another sort of 2 clusters that were quite closely related, and they were both enriched for genes that have previously been associated with activated B cells. So these were sort of our activated B cells. And then in the middle here, we sort of designated these cells as classical memory cells. This cluster here in the middle upregulates CCR7 again. But interestingly, this cluster, for example, and even this cluster, had very little or no differentially expressed genes, and were quite closely related to sort of everything. And we thought that likely indicates that they're memory B cells, because memory B cells are inherently meant to be quite quiescent, so they don't have much stuff going on. So we sort of assumed that these were our memory B cell populations. So here's the classic UMAP that you're used to seeing. We've got our atypical cells here up in purple, divided into 3 clusters. Blue represents our memory B cell populations, and green's the activated B cell populations. We had some naive cells that obviously leaked in through our sequencing, through our sorting, sorry. We have a plasma cell population, which separates quite nicely from the memory B cells. Plasma cells are quite different. And we also have a population of McGuire, we think, proliferating cells. Primarily, I'll be talking about the atypical cells, the memory B cells, and the activated cells. So, and you could do just a phylogeny of these, and we found, yes, the memory B cells cluster together, the atypical B cells cluster together, and the activated B cells cluster together, with the plasma cells clustering away from everything else. So that was all quite nice. We then wanted to see if we could integrate our 2 data sets, the SMART-seq data set and the Chromium data set, to see how SMART-seq cells fit in with this higher resolution, I guess. So this is now the new UMAP that you see when you integrate the data set. It looks essentially the same. It's turned itself around, but you still see the same clusters come out. And when we overlay our SmartSeq2 clusters on top of that, you can see roughly the majority of atypical cells seem to cluster near the atypical cells. The memory B cells cluster over in the blue atypical cells. And the CXCR3 population appears to have both activated and seems to fit in with our MBC1 population. Interestingly, and again, if we see this just divided the proportion of our 10x data sets in each of our SmartSeq clusters, the most interesting thing I think we see is that the SmartSeq2 analysis just didn't have enough cells, or we wasn't able to pull out activated cells. The activated cells seem to be found in all the populations, which we think actually suggests that that means that there are atypical cells perhaps have an activated phenotype that also is more closely related to activated cells. And again, particularly we see in this cluster 3, the CXCR3 positive cluster, and we find it's mainly made up of this MBC1 population. We also wanted to do pseudo-time analysis using monocle. So, video play. Oops, get back going here. Sorry, I can't, the videos. So we started the root of our pseudotime analysis at the naive cells up here, because this is where all B cells will start. And then you see we have, there's two, the pause function is not working. Basically, we have two. You have your memory B cells, most of your memory B cell 2 and 3 clusters, plus your activated cells cluster over here to form what we call the classical lineage, with the atypical B cells and MBC1 forming over here to form what we've not termed the alternative lineage. And it's interesting then, if we then look at how these clusters divide between the different samples, we've actually find then if you look at the non-exposed individuals who have never been exposed to malaria, they actually have quite a substantial number of cells that appear in this alternative lineage, which are related, which contain the atypical B cells. In particular, majority of it seems to be this memory B cell cluster MBC1, but they also seem to have a large number of this atypical B cell 3 subset. So it means by single cell sequencing, we seem to get much higher numbers of, I guess, atypical like cells than you would from non-exposed individuals that we normally get from simple flow cytometric analysis using, for example, CD27 and CD21. So using our site-seq total seq analysis, for example, we could look at the CD27 and CD21 expression of each of these things. And what we do find is that this atypical B cell 1 appears to predominantly to be CD27 and CD21 negative. So these appear to be our true atypical B cells, or what most people using flow cytometry would have defined as atypical B cells. But then our atypical B cell 2 and atypical B cell 3 actually have a much more heterogeneous expression of CD27 and CD21, including MBC1. And then we also have our classical lineage cells are much more likely to be CD27, CD21 positive. So it appears that CD27, CD21 negative expression doesn't appear to really predict atypical, like all atypical B cells or atypical like cells. So if we also look at CXCR3 expression, CD11C expression, we find that again, atypical B cell 1, which appear to be sort of the true or classical definition of atypical cells, appear to be predominantly CD11C positive, while atypical B cell 2, for example, and MBC1 appear to have a much higher percentage of CXCR3 positive cells that indeed are double positive. So CXCR3 is a marker that has previously been associated with atypical cells, but with this analysis, it seems to suggest that not all atypical cells actually express CXCR3. There's very defined populations. One thing we did find interesting, and it's I guess something we're interested to get from the group, is that we found when we then go and do flow cytometry analysis using these markers, we get much more defined groups compared to what we see with these total seq or site-seq antibodies. And so it seems to be these via site-seq that we get much more of a spread, I guess, rather than defined populations. And I'd be interested to hear if anybody else sees something similar, if they've done this kind of experiment before. Again, the useful thing about doing single-cell sequencing on B cells is that you can also get this VDJ data. So one thing we know, if we look at the isotype of the cells that we got, one thing we know is that atypical cells primarily use IgG3, and you can see that from atypical cell lineages. They have a greater amount of IgG3 positive cells, including this MBC1 cell. So again, sort of suggesting that this is related to atypical-like cells as part of this alternative lineage. And you can also look at VDJ mutation, and that actually showed us that atypical cells are mutated, and in the malaria-exposed individuals, they actually mutated almost higher amounts than the MBC population, which is indicative that they actually do come from a germinal center. So this sort of goes against some of the earlier reports of atypical cells that they're unmutated cells, or recently activated cells that become plasmablasts. So using this information, we then took our markers CD11C and CXCR3, as well as CD27 and CD21, and we wanted to get an idea of when atypical cells appear in the immune response using flow cytometry. So we worked with Bob Cedar in Washington, DC at the NIH, and they had a cohort of individuals who were immunized with attenuated sporozoites. So these are sporozoites that won't actually cause disease. And then every 8 weeks, they got a boost up until, so they got 2 boosts at 8 weeks apart. And then they actually received a live sporozoite challenge at 44 weeks. But we got a final blood sample just before this challenge at 35 weeks, I think. So again, we were interested in looking at antigen-specific cells in this case, so we could use our tetramers to get CSP-specific cells. And here's an example of that now. So this is an individual, this is before they were immunized. There's very few CSP-specific cells. And at week 8, just before their second boost, their first boost, sorry, there's a much higher proportion of, or there's a larger number of CSP-specific cells. And then we could examine the CXCR3 and CD11C expression of these cells to identify different populations. Sorry, here I sort of missed that up. That should be CD71 down the bottom. And CD71 is an expression of activated cells. So we can have a look at cell activation as well. So what this sort of showed us is we look at CD11C, if we divide our populations by CD11C, if they express both CD11C and CXCR3 or just CXCR3, as well as cells that don't express any of those markers but express CD71, or just even plasma cells, we can examine how the response evolves over time. So we do see that atypical cells do appear during the primary response, but majority of those cells actually, or CD11C positive cells, also express CXCR3, suggesting that these represent sort of these newly activated cells. And interestingly, after each boost, these cells all will respond again in much larger response. So they actually do appear to follow a normal primary secondary response of having a much larger secondary response. So they don't seem to be exhausted cells. And also what we can see is that after each boost, both CD11C, CXCR3 negative and CXCR3 positive populations expand following the second week, while the plasma cell population drops, which again suggests that these cells aren't just precursor plasma cells that are leading to this population. They actually seem to be a population in their own right. And interestingly, if we look at the CD27 and CD21 expression of these cells, we find that it actually increases over time. So particularly if you look at our CD11C positive cells, they start off as not having many CD27 and CD21 positive they have the majority not CD27 negative, CD21 negative, but over time and over successive boosts, they actually seem to lose CD27 and CD21 expression, suggesting actually that this might, the true atypicals that people have previously defined as being CD21 and CD27 negative may be a marker of exhausted cells, as over time they lose this expression. So if we go back to answer our question, then are they activated memory or exhausted cells? And then when do they appear in the immune response? What we actually get if we're using our single cell analysis is actually possibly all three, because rather than thinking of atypical B cells as one homogeneous population, we actually found that they make up likely a broader lineage of B cells, which we've classified as alternative lineage B cells, with some, such as the ones expressing both CD11 and CXCR3, perhaps representing an activated population. Our memory B cell 1 population, in fact, might actually be a memory population that then, when reactivated, perhaps produces more atypical-like cells. And perhaps they do have an exhausted phenotype after prolonged antigen exposure or multiple exposures, where they do lose CD27 and CD21 expression to form the sort of true atypical B cell 1 cells. And again, to answer when do they appear in the response, they do appear during the primary cell, so at least CD11C positive cells that we think are atypical cells appear. But this sort of idea of this CD27 negative, CD21 negative cell actually appears much later and develops over time, again suggesting that this idea of there being chronic atypical cells might be true, but that occurs, what that misses is this a population of actual cells that can respond that occur early on. So with that, I'd just like to acknowledge all the people in my lab, as well as bioinformatics at John Curtin, the sorting facilities at John Curtin with Mick and Harpreet, the sequencing facilities at John Curtin as well, particularly Max and Peter did a lot of the 10x stuff, as well as the Doherty Institute, the Kemery Institute. And so I don't think I have that on here, but as well, Fabio and Alda helped a lot with the SmartSeq data and helped me develop a lot of that technology, that protocol. So I think that's it for that.
Thank you, Harry. That was a very interesting and in-depth talk. I guess we could ask a couple of questions too, and then we can move to the panel discussion. And I guess Helen and I can make sure that we address everything. A comment I have on this, what I found really remarkable of what you've done is that, first of all, you show people how you can find a needle in a haystack with single cell technology, which I think sometimes it gets not highlighted as much as, oh, I can do 200 million cells all at single cell level. There is actually very important cells that are very rare, but they really matter for disease. And I think that's really nice. And so that was a comment. You feel free to say anything. And what I also found amazing is that it's kind of amazing of whatever people have been talking for years to be a population, then single cell comes in and say, actually, there is a gradient. So would you like to comment a bit on what's your opinion on what you have found broadly?
Yeah, so I think with the second question particularly, I think that's quite important, because I think one of the issues that you see is that, yeah, if you look at the UMAP, there's no single degree of cluster, and it's likely because what we're actually looking at are cells that they're transitioning, there'll be transitioning cells that can move from one population to the next. And so I think that's what a single cell I think one of the hard things about single cell is that as humans and as scientists, we really like to classify things. But I think what single cell sort of shows is a lot of the things that we're kind of interested in exist on a gradient, and by trying to label them as clusters actually makes everything, you're hiding what the real answer probably is. I think that's definitely one thing I've learned from the single cell analysis, is that, yeah, as soon as you start looking at things that are probably much more closely related, trying to cluster them becomes quite difficult as well, because we even find, we think one population that we're looking at probably could, they could all be moved into other clusters, but probably represent this weird transitionary phase. So yeah, I think that's all quite interesting. And I guess in terms of the antigen specific stuff, it's always, when you only get like 200 cells, you don't like looking at your little UMAP compared to some of the really big ones. But we did get sort of interesting information from that. And I guess if you then combine it in big data sets to see if you can get higher resolution, that can be useful as well.
I guess along those lines, Harry, we had a question from the audience, back to your idea of, continuum. So Ashwin Patel was wondering, how did you decide on the final number of your clusters of B cells, that being at 11? I think that was the experiment where you had 12,000 B cells that you were clustering.
Yeah. So that was, I mean, like this is again, I mean, I'd be happy to take advice from people who might know more about it. But my understanding is if you read like Sirat's tutorial and stuff, they talk about the resolution that you can change that does its unbiased clustering. You can sort of fiddle with that and that will give you more or less clusters. And they have like a couple of rules. If you keep on going up higher, there's like a linear, if you keep on increasing the resolution and the clusters change the linear amount, you've sort of reached your limit. But I guess the other thing is, after playing around with it, is that ended up being the most biologically relevant. We knew that they were activated in memory B cells as well as atypical cells. And from that clustering, particularly using our site-seq data, we were able to find specific differences that determined we saw that one population didn't have much CD11C and CXCR3 expression, which is sort of this cryptic population. Compared, so I guess it's a number of things of sort of how we were told to do it, but as well, just biologically, in the end, it made the most sense. So I guess that's the issue with unbiased clustering, is it's still biased by what we think is right, but I don't know what better ways there are, if there are proper real mathematical ways to do it.
Sounds legit to you, doesn't it, Fabio? I mean,
yeah, it's an interesting question. I think we can start the debate with that. It is a big problem, especially in immunology. I mean, as Harry said, and I think even, to be honest, Maggie has shown that when you started to deal with antigen-specific immune cells, doesn't matter how you do it, like Maggie has done by stimulating cells and then looking at how they cluster a couple of weeks later, or you look at with tetramers like Harry has done. I mean, the reality is that you're not comparing apple and pears. You're comparing apples with apples. So it becomes incredibly challenging to identify from the single cell data that you have cells, in this case B cells, that are actually having different functions, but they're still B cells, right? And therefore they're really difficult to tell. And those high throughput systems, they always give you thousands of genes that many of them makes a fundamental body of the B cell, right? So they're going to be the same across all the B cells. So it's very, very challenging to find which genes and really makes the difference. And Surat or any other clustering methods doesn't know that, right? So my comment would be always to be aware of the cells you're working on. I think, Harry, you said that, right? To be biologically driven, right? I don't know what Maggie, if he wants to comment on that as well in her own settings on T cells.
I agree completely. I mean, once you're in these systems, it's all, you're looking at many versions of things that are ultimately very similar. So I think, yeah, no, it's spot on. I agree completely.
But it doesn't mean that we don't need single cell, by the way, because sometimes people approach me and say, okay, so why are you doing single cell? Well, it's a very crucial thing to make clear, right?
Yeah.
Yeah. I think there is a couple of people actually that have asked questions.
Yeah. Is it okay if I invite, we've had a hand raise and a question from DU, who's one of our interested researchers from Queensland. If you wanted to chime in with a couple of thoughts, Dee, you can, I think, speak.
Oh, I just have this technical question. As Harry just used, most people still use a 10 times genomics platform. And so just wondering, for the BD respiratory platform, what's the number of cells is recommended? Because for some small population or small cluster, the number of the cells captured for each run make a lot of difference.
Yeah, no, it's a good question. So our cell loads, we go down to as low as 100. But I think, as Harry said, I mean, if you have 100 cells, you know, use something like SmartSeq where you're going to really be able to really look at those 100 cells, because that's a much better platform if you're down at those cell numbers. But in the upper bounds, a lot of our users are routinely loading 20,000, but we're starting to see people push that much more often up into 40 to 60,000. We have much less data testing our system in that range, so we can't speak to it as faithfully as we can in the 20,000 range, where we just see, you know, everything's beautiful, but all the data sets we've seen so far are beautiful up in that range as well. And we don't really see any change in, you know, cell capture rates or anything like that. We're still seeing about 75% on average, probably, of cells from, you know, cell load to what you actually get in sequencing, which is, I think, pretty similar to 10x. But yeah, I think that the microwell system does allow you to really push those numbers pretty high.
Yes, from some of the BD information brochure or the introduction of the platform, the capture rate will be about 60 to 70%, or if I remember it correctly. So if you load 20,000 cells, do you mean we can get 10,000 cells for sequencing?
Yeah, so I mean, it really depends on the cell type. So, you know, internally we're loading often cell lines. You load 20,000 of those, you're going to see easily 15 to 17,000. When you go into PBMCs or even more activated or more delicate cell types, you're going to lose more. So then you are probably looking at, you know, if you load 20,000, you're going to see closer to, I don't know, 12, 13,000, something like that. So yeah, that's a good call out, that it really does depend on the cell type. And, you know, you should adjust your cell loading accordingly.
Okay, thank you very much.
Yeah. Maybe I can comment a bit on that, Maggie. I've done a little bit of work on that. The Rhapsody, I think it's a great way. Let me put two things. If you are looking at cryopreserved samples, it doesn't really matter if you do 10x or Rhapsody. I mean, cryopreserved samples, you always have a bit of a less recovery than fresh cells or cell lines. But I think that 50, 60%, it's a good ballpark in general. We tried on cryopreserved samples across different cohorts. So, and it really depends how people cryopreserve samples and the quality and the age. But I would say, not to be, you know, respectful, it's the truth. There's no major difference in terms of cell recovery between 10x and Rhapsody. The interesting thing, the comment, I guess, is also part of what the next question is asking is, I think that one of the good things of Rhapsody is that you can be flexible in terms of numbers. I think, Maggie, did you say that you're seeing loading like 40,000 cells on the same run?
We're seeing 40,000 pretty regularly at this point, and a couple people have gone up to 60.
And do you think it's because we always been cautious about, you know, learning from the 10x where you have a much tighter numbers to respect, right? For Poisson distribution and also for issue for clogging.
Mm-hmm.
The Rhapsody doesn't have basically a pushing, right? A pressure that brings these cells at high speed there. So it's much more gentle. So do you think that is a quality of this machine that could increase the flexibility of what you could do in cell numbers and things to do?
Yeah, I mean, I think absolutely, because there isn't that microfluidic aspect, you have to worry a lot less about kind of the cell suspension going in terms of, you know, do you have a ton of cells? Are they weirdly shaped? Anything like that. So yeah, I mean, I think it does help enable that. I do think, though, you highlight a really great point, which is that, and the flow people have been saying this, right, for ages, crap in, crap out. You know, it all depends on what you do upstream to get that single cell suspension, and whether you're using Rhapsody or 10x, you know, the cells going in have already undergone a lot oftentimes. And so I think that's an area where I'd love to see more about, you know, with different tumor dissociation methods, with flow sorting, with all of these other things, how do those things specifically impact, you know, Rhapsody? It's nice because we are just pipetting it gently into the microwell, but they do have to settle into those microwells. And so there are aspects of cells that might, you know, if they get fluffy, if they get something like that, they aren't going to settle as well. Whereas with 10x, it's going through another droplet formation process. So there's different kinds of things that are going to happen to the cells with all the different preps, that some might do better with 10x and some might do better with Rhapsody, if I'm being fair to both platforms.
Absolutely, yeah.
We had a question on Twitter from Piero Carnini, who had a question to Maggie, if you could elaborate a little bit further in terms of Rhapsody's TCR, BCR sequencing, with that statement you made about what versions or any versions of random primers and depletion of the RNA before sequencing, if you could kind of speak a little bit more.
Yeah, so we don't do any, so the random priming question, we use like a primer panel, which should get all of the V segments, and that includes for gamma delta cells, not just alpha beta, for TCR, for example. So this primer panel should kind of get everything. And then, yeah, so there's no random priming. In terms of the rRNA depletion, and that's a great question. I mean, for any of these platforms, the percentage of reads going towards ribosomal is, that's useless. What we've seen interesting with the Rhapsody is that we have fewer reads than other platforms going towards ribosomal RNA, but we have more reads going towards mitochondrial. And so the mitochondrial is, a lot of researchers use that mitochondrial cutoff as a cutoff for live versus dead cells. And what we've seen is with Rhapsody, you have to move that cutoff up because we do just get more mitochondrial background. We think that's because our lysis buffer is very severe. We want to get that lysis buffer in there. We want everything just to explode open immediately. Droplet systems need to be very careful about the lysis buffer, because they have to keep that droplet formed. And so it is a much more gentle lysis buffer. So we think, that might be one of the reasons that we're seeing, more mitochondria just bursting, is because of that more severe lysis buffer step. So sorry, that's kind of an extension of the question, but we don't do any ribosomal RNA depletion. We don't actually see that we need to with Rhapsody. There's very little rRNA signal.
Great.
Perhaps again another comment, because it's interesting to hear this thing. So the key word, in my opinion, is again flexibility, right? Because it's such a simple machine. Do you envisage to actually see users be able to play around with this? For instance, do have depletion, right? Or do add other techniques, right?
Oh yeah, absolutely. No, I think it lends itself to a lot of flexibility. I know there's also there are researchers who are saying, you've got those magnetic beads. I don't necessarily want to use the Rhapsody, but I want to use them for a bunch of stuff in my own well-based system or something like that. So I think also those beads themselves lend a lot of flexibility even outside of the Rhapsody. I'm not advocating to use them outside the Rhapsody as a representative BD officially, but we're hearing a lot of uses for them, so that's exciting. And then yeah, sorry, I can't remember where else it was going, but yeah, I do think there's a lot of flexibility.
I had just a comment to Harry that I really enjoyed your presentation, and the fact that you were able to reflect on, if you had done it again, you would have, you've learnt lessons. And I think that honesty and presenting that type of information to the community is like just so phenomenal and stellar. So was there any extra little tidbits along the way that you had to share?
No, not exactly. I mean, like, I guess the cell prep, I think, is the most important bit. We thought we would have gotten more cells out of it from the cell numbers we got. And then when we talked to a Chromium representative, and they talked about how they always get much higher cells coming out of things. And then, and he said that most of the problem is that people don't count their cells well enough before that they put them in, or there's issues with cell counting. And that's why you might get lower cell yields than that 50%, because you just didn't actually put in. So we've done it a couple of times now. We've made sure we count the cells a lot more before putting it in, and make sure we're pretty sure what's going in. And I guess the other thing is I don't know what everybody else is doing, but I know like John Curtin is, particularly now that there's all this hashtagging stuff with SiteSeq. You can actually share, because I think the biggest problem with single-cell seq at the moment is just how expensive a single experiment is. And in terms of experimenting with anything, it's kind of scary to do that when you're spending so much money on it. But if you can, I don't know much about the Rhapsody, but with Chromium, you've got those 4 lanes. But if you can put, with cell hashing, if you can put multiple samples in each lane, there's possibility of collaborating with a lot of people on this sort of stuff, which I think is kind of an important way of doing it, because it just makes everything cheaper and easier to do.
Yeah. And I guess I'm a cytometrist by trade, I guess. And so I was kind of curious whether, now that you've gone ahead and done all of that single cell transcriptomic work, have you now redesigned your cytometry panel to better define those lineage?
So that's particularly what we wanted to try and do, and we have been doing that. And now I'm about to leave the lab, but other people are sort of using that data set now to focus on downstream projects. The one thing we did find is that it was, in particular with our stuff, is that there actually wasn't many good surface markers that delineated any of. So in the end, it almost sort of told us that single cell was actually the best experiment for this, because between some of the populations, there just wasn't very many surface markers that actually differentiated them. So you wouldn't have any flow panel, or that we can see at the moment. No, there was not really any flow panel that would have pulled them out. And so I guess this is the power of single cell, is that there are, there's all this stuff hidden underneath the surface that lets you tell the difference between some cells that you'd never be able to tell using flow cytometry.
Yeah, sure.
I have a question for Harry, actually.
Mm-hmm.
So on your comment about, that you saw less definition with the site-seq, how did you set the boundaries then for positive versus negative? And yeah, I guess I'll start with that.
Well, so we used that naive cell population to come in, because we knew that they should be naive to CD11C and CXCR3. So we ended up using that kind of, I guess, as not a fluorescence -1, but as a kind of a negative control, I think, to base it. But that is one thing that we found tricky, is we weren't quite sure where the negative and positives should be. So yeah, that was a bit hard.
Yeah. I think with the antibody oligo technology, this is something, it's so new, and we're all still learning how to analyze the data, and there aren't a lot of negative controls in a lot of experiments, right? So for you have those naive that you could use to set the gates, but there are also a lot of, a lot of experiments where you don't have an obvious negative control. And so we're talking a lot internally about how to help you, generate data to help understand how to set these positive and negative gates. But it also is so much related to, the metrics of sequencing. How deeply are we sequencing? How many reads per cell? What's your sequencing saturation, your sequencing depth, and things that we're not, aren't often presented upfront in a paper, that if they're presented, depending on those numbers, the entire resolution of the data looks so different with sequencing. It's like changing the voltage on a flow cytometer, right? You change your sequencing, it's the same thing as changing your voltage. Everything looks really different. And so that's one thing that I am excited as the field is shaping up. I do feel like we're going to start having more of those conversations of how do these really basic metrics and basic parameters of the data change how it looks, and how can we change those to get the better data? So
yeah, well, I think because that's one thing we found, particularly with the site-seq stuff, is that normally if you're doing a flow cytometry experiment with a new antibody, you titrate it and you check it, and but you can't really do that with any of these without running a whole nother sequencing experiment very easily. So I guess the hardest bit about that is we sort of, in the end, the reason why we didn't put too many in there is because we didn't know exactly how it would work, and not many. We bought them just as they sort of BioLegend released them. So we didn't know exactly how they were going to work. So that's one of the reasons we didn't actually include that many markers, because we didn't want to screw it up by having all this dodgy. But we think in general, we think it worked quite well. There is, but again, it's this issue of, rather than seeing distinct populations, we appear to see shifts and stuff, and we're not quite sure if the double positive populations are really double positive, because we don't see that in the flow cytometry data. So we're not quite sure if we believe all of it. But I guess it's hard then when you're showing this to we're trying to publish it, and the reviewers are asking, why does this look different to that? And we're going, we don't really know.
I think Harry, you make a good point. Just to conclude this, it's quite interesting. I just want to say that, as Maggie said, I think with the increase of these oligo antibodies, while I understand what you said, if I go back, I buy a few more, but there is an interesting point to make here, right? That the unpredictability of where to put the threshold like Maggie said, you might have the function of cell count or coverage or the quality of your sample. Like we have been working on a lot with 156 antibodies, okay? And I can tell you that it's becoming challenging to even monitor them. And each of them have a different threshold. So why you have 4 or 5 antibodies? So I think it's going to be really exciting. And I think it's going to end up to the end of manual gating, in my opinion. And it's going to go into machine learning era. Yeah.
Great.
Anyway.
I think that's a really nice place to finish up our conversation, that, you know, we're all excited towards the future. And so with that, and we're basically out of time, I just wanted to thank our speakers once again and thank my co-host, Fabio. So this has been a really wonderful episode. So Maggie, your countrymen are a little bit busy next week with the Sutton election. Wish you good luck on that. But with 1 week break, we will reconvene again on Friday the 13th for a continued celebration of single-cell technologies in immunology research. There's just so much going on that we couldn't jam pack it into one episode. So we're really excited to welcome a whole range of really great speakers. So we've got Professor Helen Rothenberg joining us from California. We've got Dr. Ash from Melbourne, and Joel as well, a recent postdoc down in Melbourne as well. So this is going to be an excellent episode. So please put it in your diary, lock it in for Friday the 13th of November. And another date for your diary is our online poster session. So those of you, obviously you're here and now will receive an email later this week with more detailed information in terms of registration. So we'll get that later this week sorted. But in terms of your diary, if you put a placeholder for the week of the 13th of November, that will be fantastic. So again, encouraging those of you who are familiar with OzSingleCell, we have a wonderful celebration tradition of the Krisny Awards. So I encourage you to get your applications entries ready for that. And with that, I'd like to close out the session. Thank again all of our sponsors and everyone for joining. Thank you.
Thank you.